Good afternoon. This is Nelly Poyak from Dana Farber. Welcome to the FHCC Leukemia Lymphoma Society Connect Science Seminar Series. And a reminder that the talks will be posted on the website, the FHCC Harvard EDU website, the slides, so you can look at it later on as well. And also, as a reminder for everyone, please submit questions in a Q&A function on Zoom, and we will try to answer as many questions as we can at the end of the presentation, and the speaker will have access to the questions and she can get back to you later on. So with that, it is my distinct pleasure to introduce today's speaker, Dana Farber, who is the chair of Computational and Systems Biology program at Sloan Kettering Institute. She's also endowed Ellen and Sandra Gary Chair and director of the Ellen and Sandra Gary Metastasis and Tumor Ecosystem Center. Today, Dana will talk about the dynamic view of the tumor ecosystem. Dana, thanks for presenting.
Hi, everyone. Thanks, Nelly, for inviting me, and thank you, Dana Farber and Leukemia and Lymphoma Society for really organizing such a wonderful series for the community. I'll begin with my disclosures. And basically, I have no financial interests, and mainly I'm disclosing that I'm simply a strongly opinionated person, and you'll probably see that throughout the talk. I decided that this talk, in addition to telling you some scientific stories, I'll also take an opportunity to do a little bit of education in computational biology. It's become really easy to collect single cell data with the 10x machine. It's very, very easy to put sample in and get out single cell data with different software tools such as Seurat. You can push buttons and come up with pictures that are worthy of placement in New York's Museum of Modern Art. And that has left a little bit, some bad practices in computation. One of my pet peeves is overreading what I call TSNE tea leaf reading. TSNE, and there's nothing special about TSNE. Same thing goes for UMAP or force-directed layouts, or basically any attempt to take data at 20,000 dimensions and look at it in 2 dimensions can lead to a lot of over-interpretations. It's very powerful visualization. It can trigger ideas. It can trigger hypothesis. But please always look at your data back in the high dimension when trying to actually make a final assessment. One of the problems, beyond the fact that just taking the data from high dimension and putting it into 2 can create a lot of misleading information, is every step in the data processing matters. Even how to normalize a single sample, you have many, many choices that can have big effect. Feature selection, clustering, how to correct batches across different samples is one of the most challenging problems, which as a field we haven't solved. And unfortunately, I don't have a single solution to give you. There's no best solution. It really depends on the data and what downstream analysis you're going to do and what question you want to ask. So to dig in, I'll just spend a little bit. I can't go over each of these choices because that would take the entire hour. Just clustering. Clustering is what we do a lot. We like discrete objects. We don't want to deal with 100,000 cells. We'd rather deal with 50 clusters. Clusters is the type of object that sort of typically matches to the cell types that we know and love, and it's a lot more easy to interpret our data once it's organized and decomposed into cell types. And one of the most common methods for clustering is a graph-based method. Basically, you treat each cell as a node in the graph, connect cells to each cell in the graph with an edge to its most similar cells. So you have some type of similarity metric, how similar their expression vector is, and build nodes. And then, we take methods from social networks, and such to identify communities. Highly interconnected nodes, and these usually match cell types. Now this recipe, which we first showed that this is a good way to cluster single cell data, still with CyTOF data together with Gary Nolan, and has three components. How do you build the graph, and particularly what similarity metric you use? How do you perform the density and estimation and break this graph that you just constructed into clusters? And of course, some of the most challenging part, now that you've clustered it, how do you label and interpret your resulting populations? And I'd like to bring your attention to what I think is pretty important, which is the Jacquard metric. Many of the methods that have implemented this, many of the common online tools everyone uses, or interactive tools, such as, for example, the Seurat tool, does not use the Jacquard metric when building this graph. And I actually think this is pretty important, and I'll give you an example why. So the Jacquard metric, what does it do? Basically, it says, build your graph in any way you want. And because this data is so spurious, you can see these two clusters, this one right here and this one right here. Single cell data is not nice Gaussians. It has very complex shapes, and it has clusters of very different sizes and shapes. And there's almost no way to disconnect these two clusters. But then using the Jacquard metric, which basically says, once you've built your initial graph, and I really don't care if you use correlation or the Euclidean norm or any other norm, also doesn't matter how many neighbors you choose. Now you clean up the graph and you sort of induce additional modularity by saying your weight, how similar I am to you, is how many neighbors we share. So basically it counts, tell me who your friends are and I'll tell you who you are. And this is really great at doing all sorts of things, such as removing spurious edges, really captures very small populations very well, really scales with local density. One of the biggest computational challenges in single cell data is we have populations with different sizes, T cells versus progenitor hematopoietic stem cells. We have really dense regions of many, many cells that are similar, and much rarer cells. And as you know in biology, sometimes the rare cells matter. And what I like about the Jacquard is it accounts for these density distances, and it's very, very robust decay. So just to show you an example, here's the typical result that you'd get out of most algorithms that you run these days without a Jacquard, such as in Seurat. And Phenograph, which is our implementation that includes simply a Jacquard. That's the only difference. And you see that, in some default parameters, these are pretty similar. Again, here we found 11 clusters versus 13 clusters. Again, Phenograph is very good at finding very small clusters. Does anyone think that Tregs are an important immunological subgroup? They're pretty small. But the real difference comes in the robustness. So there was a choice in parameter. Here it was one parameter choice. Now what happens is you change this parameter choice or change the data. Without a Jacquard, you can see you go from 2 to 114 clusters, and this doesn't look very stable. In Phenograph, because of the way the Jacquard works, it's actually incredible robust, and we get very similar answers no matter what number we get. So this is the reason I really think it's important, all these tiny choices in how you cluster. Now, where does it matter most? It really matters the most in these T cells. This population really cares about these choices. So a lot of single cell analysis is done on neurobiology. And here's an example from my friend Aviv's lab. And neuronal cells, they really separate. The mature neural cells have very distinct cell types. You can use any method under the sun, and they will separate quite nicely into very distinct clusters. But here are two different groups of T cells. Here are T cells in breast cancer, and here are T cells in lung cancer. And you could see that it's more blob-like. And while I told you never to overread a T-SNE map, I can tell you that also when you go into high dimension, the T cells are a lot less well separated, have a lot more continuities. And, people have been using a concept called diffusion components. You've probably seen these in many papers. So just to sort of show you what diffusion components is, diffusion components assume that the data creates some high dimensional embedding. So you can imagine the Swiss roll. And while the Swiss roll is embedded in 3D, it has an innate 2D structure in it, the sort of spiral of the Swiss roll. And the first diffusion component will get the spiral will get the longest proportion of, the longest axis of variation within the embedded object. And then the second vector will get the fact that this is a ribbon, that it has some width. In a way, you can, you know, view diffusion maps as sort of a nonlinear version of PCA that really tracks data density, where you see, you know, PCA would really mess up in this type of data, and this sort of nonlinear component that allows to sort of swirl around with the Swiss roll is really what you want. And diffusion components have been very good at tracking development. Here's, you know, our work in looking at some T cells, and we really see T cell development from, you know, the double negative cells in the correct order, all through the double positive and the CD4 and the CD8 cells, and the diffusion components really tracks maturity and development. Of course, when we made some of the right early choices in how to process this data. And going back to T cells, indeed, T cells have a very continuous nature to their single cell structure and geometry in space. You can see quite a few diffusion components in most T cell data sets. You'll see that the first diffusion component is often a signature of T cell activation, sometimes T cell exhaustion or differentiation. Here, the first two components are activation and differentiation / exhaustion. You can really see a gradual, continuous nature. There are other components, most of them to do with metabolic gradients, hypoxia. If you go a little further down the line, you'll see sort of energy metabolism. But you really see that there seems to be a number of components, a number of gradients that really hint to a very gradual, continuous nature of the data rather than distinct cell types, which we like to slice and dice the data. And here I'm showing data from CD45 TILs from breast cancer patients in a work together with Sasha Rudensky. Again, because the T cell activation signature is repeatedly one of the strongest diffusion components, if not the strongest component of variation you see in T cells. So one of the things we ask is, well, could the TCR, could different TCRs, which, you know, account for all this variation, is the reason that we see T cell activation over such a wide gradient actually a combination of the many TCRs, each of them having a much more narrow gradient. So, you know, we use TCR signaling combined with single cell RNA-seq to address just that. And we can get both the TCR as well as the activation status from the RNA. And what we saw is, yes, when you limit your data here to each clonotype separately, so each of these boxes, so this blue box is all the T cells together, and here these little green boxes are each TCR separately, you see that there's differences. Some TCRs are much more activated than others, but we do see that within each clonotype, there is still a very wide range of behaviors, a very wide range of activations, where the TCR can, depending on the patient, we did 3 different patients here, can only explain between 30 to 50% of the variance, and the majority of the variance is not explained away by the TCR. And moreover, this wouldn't explain all the many other diffusion components that are really suggestive of metabolic gradients related to inflammation, hypoxia, as well as many other metabolic features such as energy metabolism. Okay, let's dig into a little bit more about immune subsets. It's not only the T cells that have such an incredibly rich structure in the data. Remember I told you that sometimes some of the rare cell types are very important. So this is data from the tabula amoris from CZI, from Steve Quake, looking at the mouse spleen. And you see here the dendritic cells are marked. You can barely see them. There are 3 green dots right behind these dendritic cells. And, you know, a lot of machine learning algorithms, they say that where most of the data, that's where the most important thing is. But, you know, arguably most people, certainly my friend Yura Khan would say that dendritic cells are quite important. Now, together with Sasha, again, we decided to enrich for these D cells and, you know, dendritic cells and sorted them by facts. And again, just to confuse you, there's diffusion components, which are DCs, and then there's dendritic cells, which are DCs, just to, you know, further confuse you. And, you know, once we've sorted them and enriched and got many of them, you can see that the dendritic cells themselves are a whole world onto themselves. And there's much variability and much structure and many subtypes. In this particular paper recently published by Chrissy and Herman, we identified a very important role for Tibet in defining new subtype status for some of these dendritic cells. Let's look at these dendritic cells again. Subtypes aren't exactly clusters. Here's a phenograph clustering of these subtypes, and you see that when a biologist sort of maps it based on their favorite markers, you see that it's not a complete, perfect one-to-one mapping between cell types and clusters. That is because the cell types are often based on surface markers, and the clusters are based genome-wide on everything that's changing, be it cell cycle, be it all these metabolic signals and that I was talking about, and all sorts of other biological processes that might happen inside these cells. And the clusters is also not the be-all, end-all. If you sort of look at the landscape, if you look at the contour plots of the data density, you see that even within each of these clusters, there seems to be more shape and more structure, and the density isn't, again, form a nice uniform Gaussian ball where most of the density is in the middle. So if we sort of want to zoom in and we sort of decide, let's zoom in here. And remember, a TSNE or a UMAP can only use as much as the landscape that they get. So here, what we have is we zoomed into this little component right here and see that there's incredible additional structure here. And we actually can distinguish 4 different states. And these different states, why are they different states? Well, they're defined by different gene programs, different patterns of co-expressed and co-varying genes. We're here in the pink cells, we have gene program 1, where these genes have a very strong structure of co-variation and a relationship of expression. Whereas the green cells right here have a completely different gene program involving both different genes and a different relational structure, a different covariate structure between the gene. And when I sort of use the curse word covariant, which some biologists don't like, when I use, the bottom line is that it is these sort of plots here that show you the gene-gene relationships and how the genes statistically relate to one another. And this covariation really captures the biology underlying these cell states. These cell states are driven by regulation. All of biological states, the clusters that we see, the structure that we see in the data is driven by careful regulatory mechanisms that regulate how a cell should behave, what cell states are possible through an intricate regulatory circuit. So we want to move on to the next step, and remember, this covariation patterns is going to be critical. Everything I've shown you till now, it's all complications in analyzing a single sample. Things get much more complicated when you want to analyze multiple samples, and this gets really bad when you want to work with patient samples. If you work with mice, they're really friendly. You can do it in a very controlled way. You can control exactly how and when and what you do with the sample. When you're getting patient material from the OR, there are so many factors that you cannot control, and therefore, sometimes the messiest data, the strongest and hardest batch effects that I've seen come from samples in the operating room. But of course, we want to be able to study samples directly from human. We want to make it as relevant as possible and see what is happening inside humans, and single cell analysis makes it possible to actually look at the tumor immune ecosystem inside, human patients. Again, this is breast cancer TILs as before, but now we have 4 different patients, and every dot on this map is colored by the patient, and you can see very strong patient effects. And the question is, how much of this is technical artifact and how much of it is biology that matters, because there's just so many technical things that happened along the way that can create so many technical artifacts. One of the signs that I like to see is, the entropy of the sample. Basically, if you go back to that nearest neighbor graph I described in the beginning, I want, in a well-mixed sample, that most of my neighbors come from different patients. So here, most of the orange neighbors come from the orange patient, and that's a low entropy area. Where high entropy means that, I could be from one patient, but many of my neighbors come from other patients. And the problem with batch correction, it's something we haven't solved. All batch correction algorithms make strong assumptions. They all have strong trade-offs. They don't have any free lunch. Many of them really try and force similarities that might not be there. So they assume strong similarity, where often our samples have a real biological difference. Some of them, in order to normalize, break that covariate matrix, which I showed you earlier, which shows the gene-gene relationships, which we care about. In fact, way too many batch correction algorithms are linear and break relationships between genes. And so globally, we see a very low entropy here, and that worries me. So we decided to try and develop our own batch correction and clustering method. Another problem with global normalization and batch correction is that different cell types have naturally different numbers of genes in them. A macrophage is an RNA factory. It has hundreds and thousands of transcripts in it, truly biologically, whereas a naive lymphocyte, a naive T cell, has only a couple thousand samples, and many of the normalization methods try and equalize them, try and assume that they have equal library size, and this can really mess things up, particularly in differential expression analysis, when you make an assumption that the macrophage and the lymphocyte have the same number of transcripts, or should have the same number of transcripts. So we felt that certainly for something that has cell sizes that are as different as immune cells, we want to be able to normalize each cell type separately. John Marioni's SCRAN does that as well, and really for tissues that are very varied in the number of transcripts per cell, it makes a lot of difference. Another thing that we did in BiSKIT, and this was sort of spearheaded by Elham and Sandhya when they were both still in my lab, was that we really want to preserve these gene-gene relationships, that we really make that covariate matrix, the gene programs that are on and the relationship between genes and the data, that's what we want to preserve as best as possible across samples. And our key assumption here is that batch effects, are mostly independent of this biological structure. Batch effects affect genes, but don't affect programs in a way, because programs are a biological entity. And so BiSKIT is an algorithm that aims to identify these sort of covariate structures in the data, assume that these covariate structures are real, and then correct the data to sort of clarify these covariate structures in the data. And these covariate structures also help you define the clusters themselves. And so what you see, this is the same data on the left and on the right, before and after BiSKIT normalization. Now you see a much clearer mixing of the cells. You see that the cell types have more structure to them. When you actually label the cells and find out what they are, it also makes a lot more immunological sense. And this entropy down here is a lot more mixed. And BiSKIT is very robust. You leave one eye, you can mess it up. I really like algorithms that are robust, as I showed you earlier from Phenograph. And because it's a parametric model, you can actually directly pull out differentially expressed genes directly from the parametric model itself. One of the advantages is that BiSKIT is computational heavy. And as much as, for example, I like applying BiSKIT to tumor immune cells, because these are healthy cells, and I assume that their underlying circuitry is a healthy circuitry that's similar across all patients. I wouldn't apply it to cancer cells, because in cancer cells, it's exactly the circuitry that goes wrong, and it's exactly the circuitry that can go wrong in very different ways across patients. So this is something, again, that's why I said that each algorithm and each method depends on the data, the question, and the assumptions you're willing to make. Unknown caller. One of the things that we see is that the covariation structure can really pick up some really clear biology that you would not pick up if you would look only at means. For example, here we have Tregs, and these Tregs have very similar mean expressions, but very different covariate structures. Here's two examples. And this covariate structure actually changes in many of your favorite immune surface markers, such as TIGIT and CD27, where in some clusters they're correlated and some clusters they're not correlated. So they're very strong covariation patterns that can define cell subsets. And we confirmed this in site as well. Again, they're gene programs and cell types, and they're different things. Covariation really helps us preserve signal across technologies. So here's another example. And the original data for the paper we collected while I was still at Columbia using InDrops, which is a 3' technology, with 8 patients that were shipped from Memorial Sloan Kettering to Columbia. One of the big parameters is taxi time and New York City traffic. And then after I'd moved to Sloan Kettering, we took 3 new patients. We sequenced them on 10x with a 5' kit. That's where we got the TCR I was talking about. So we have 2 very different technologies which we processed independently. But we could get a nearly 1-to-1 mapping of these clusters. This we couldn't get when we applied other methods, but because we really searched for these covariation patterns, and because these covariation patterns reflected something real in biology, whether we use this set of T cells collected from these patients in that technology, or a completely different set of T cells in a different technology 3 years later, we got the same underlying covariate structure, which allowed us to get almost a perfect 1-to-1 mapping with the T cells that we found, really stressing how important this covariation is to really get robust signal. Okay, this is all unpublished work, but I wanted to take that little foray into how I think about data analysis and how people should be careful and not just press button and think about how am I analyzing my data and how does it impact the conclusions I'm making. So what I want to tell you about is some of the things I'm thinking about a lot lately is how do you model response to therapy? How do you model generally dynamics and longitudinal cohorts? How do you model things that change over time? And Biscuit is, by the way, in responding to a question, Biscuit is available, but it's very hard to run. It's heavy and requires, it's hard to make it user-friendly, but it is available, not necessarily on the cloud, but it can be downloaded from my GitHub, I believe. But again, back to the biology, we want to understand how populations change, how their transcriptional states change, what differs between responders and not responders, what matters. We really want to use this longitudinal data to find the differences and find the differences that matter. And there's a lot of challenges here. So, here I want to tell, I really often like telling stories that I did together with people in the institute I'm talking with. And this has been a wonderful collaboration with a dear friend and colleague, Kathy, and her postdoc, Pavan, and Elham, who's now independent at Columbia, but was my postdoc. And it was really a wonderful collaboration between, of course, mostly Elham and Pavan and Kathy and I as well. And again, one of the reasons it was so wonderful to work with Kathy on this project and Pavan is that, longitudinal data is challenging enough. And you really, first step to have a chance to attack it is you really need a well-designed, well-annotated cohort where your samples are matched. You want to take as there's so much patient heterogeneity. You want to take the confounders out of the picture as much as possible. You want, really high quality samples. Kudos to Kathy for preserving her samples and developing protocols to get data at incredible quality. So we didn't have to deal with bad quality just by the regular challenges of dealing with patient data. And of course, you also need to have good modeling approaches. And, this is a collaboration where, good data, good clinical samples, good clinical understanding was married with what I believe, in my humble opinion, to be good analysis. Anyways, it was all Elham's idea. So, I can give Elham some compliments. And, here's what we got. So what we decided to study is, perhaps one of the oldest forms of immunotherapy, the donor lymphocyte infusion. So this is, after bone marrow transplant. Sometimes some patients relapse. This is in the case of CML. And, sometimes you can reverse this relapse by infusing donor lymphocyte material, specifically CD4s from the donor. And so, this is something that Kathy and Pavan had been working on long before we decided to sort of join in. And they, as a starting point, I've done a serious analysis of bulk data and sort of figured out that one thing that distinguishes between responders and non-responders is that the responders seem to have many exhausted T cells pre-therapy, and post-therapy, sort of you get a reversal of this exhaustion. But we really wanted to understand that much deeper. What are the T cells that are mediating this response and this resistance? And that bulk analysis gave some insight and some satisfaction, but we wanted to go deeper with the power of single cell. And for this, Pavan really put together a beautiful cohort. And we had multiple time points for some patients, but really for every patient, we had a sample shortly before the DLI, as well as, at a later time point where it was clear and, having a very binary response in this case of either responding quite strongly and quite clearly or not responding. So we had sort of a post-response time point. And for a reference, and this was actually pretty important, at the beginning we used healthy bone marrow, but we realized that that's a really bad reference. So the reference we ended up correcting for is just patients that never relapsed. So patients that had received the transplant and stayed, happily in remission. So, we ran Biscuit on it, and the first thing, again, you get clusters and you want to interpret them. And like always, when we analyzed T cells, we found a really, really rich landscape of many T cells, many different clusters. And one of the thing I like to see in clusters is that the clusters weren't unique to each patient, that each cluster had a very, very rich landscape of different, each cluster really liked to fall in multiple patients. And you can see that right here, that every single cancer, the cluster has sort of multiple colors, each color representing a different patient. Now, while the clusters were mixed, they weren't uniformly mixed. So they did have a structure to them. And, there was definitely clusters that were enriched for pre or post DLI, and that really mattered. So you could see in this map a very strong structure here. Then you also see that there's responders and non-responders as well that create structure in the graph. And so we wanted to dig in deeper. So we saw that the clinical covariates that we cared about, had structure. And first, we wanted to see, can we annotate these structures? And so the typical thing to do is sort of map between signatures in all these clusters. And we saw that, yes, the clusters had many different subtypes, including some very rare subtypes. So you could find all the important immune subtypes among the clusters. They had many differentiation states from naive all the way through terminally exhausted and different types of memory states. In addition to the sort of classical surface marker based classifications, the clusters, as we had with the lung, also, differed greatly with all these sort of metabolic and immune programs, whether it's energy metabolism, senescence, as well as different interferon and inflammatory signals. And so, each cluster we could associate with many different signatures and many different gene programs that were relevant and active in that cluster. But, this is sort of annotating the clusters without any relationship to the clinical covariates. And we really collected this data to try and understand the difference between responders to responders. So we first tried to ask, well, what underlying factors can explain this diversity in terms of responders and non-responders? And using all the standard toolkits that we use, again, the first tool, my typical first go-to is the fusion components. The fusion components explained a lot of the major components of variation in the data, but none of that separated responders and non-responders. Some of the strongest signals in the data really seem to be signals that are do not separate responders and non-responders, which is what we wanted. We also tried principal components as well, and you see a lack of separation. And so, here's where Elham had a great idea, and she said, well, there's so much, patient-specific factors. There's so many strong biological processes that dominate everything else. Can we find some common factors that explain a lot of the variation in the cells? So instead, we decided to run common factors, which says, after you explain away some of the unique factors, can we find a small number of common factors that explain, as right here, we're trying to explain as many different cells as possible and to capture the sort of joint changes or coordinated changes of as many cells as possible in the data. And when you sort of push your algorithm and say, hey, algorithm, what I want you to look for are these common factors that are shared across patients and shared across as many cells as possible. And that's what I want you to find. Really putting less emphasis on specific patient variation. When that's what you ask from your algorithm, that's what you get. And we indeed now got 3 dominant factors that explained about 75% of the data. And these factors really varied. So factor 1 explained and separated the responders in the blue, and factors 2 and 3 sort of matched the non-responders. So now that we had these factors, we could say, okay, what is the biology of these factors? What is the underlying biology of these 3 factors that our common factor analysis just found? And so factor 1, the factor that sort of matches the responders, again, it caught what Pavan and Kathy had found earlier, that exhaustion terminal differentiation signature. And this is, again, looking, and then, quite a bit of other things that relate to immune signal. When we looked at the factors 2 and factors 3, we saw many different, dysfunctional modules. We found very different reasons that each patient didn't respond. And what I want to bring your attention to is that often when you have these gene signatures, there's high overlap between the genes. But in this case, almost each gene signature had completely different genes, really showing that while all happy families are alike, and they all sort of share this sort of terminal exhaustion immune activity phenotype, the unhappy families are unhappy each in their own way with many different forms of dysfunctionality that lead to perhaps this lack of response. Another really surprising thing that we found is actually the responders had a lot more heterogeneity, a lot more variability up front. So one of the metrics that we like to use of how much variability there is phenotypic volume. So if you think of all the possible phenotypes of T cells, and if you look at all the possible combinations of programs that can exist and all the combinations of cell states that we see in our data, and just how large the space of possible phenotypes is, we can quantify that with a phenotypic volume metric that we developed. And we see that the phenotypic volume in responders is order of magnitudes higher than the phenotypic volume of non-responders. So there's a sort of plasticity, richness, diversity in the cell types up front, even pre-treatment. So can we identify these relevant clusters that can really predict this response? We connected it to factors, we connected it to variability, but can we drill down to these clusters I was talking about earlier? And here we focus on just looking at pre-treatment. And our analysis found 4 such clusters. Now, what's really important here to state is that because these clusters are of varying size, in order to get a robust signal, we had to really take into account that each cluster has a different size and each patient has a different size as well to do sort of an unequal variance t-test to get these 4 clusters. And what's unique about these 4 clusters? These 4 clusters are all terminally exhausted clusters. They all had a very strong p-value for a terminally exhausted signature that Pavan derived from mouse. But not all terminally exhausted clusters were predictive, only a certain subset of these. We asked, okay, this is all preclinical, but we had pre-DLI. We asked, okay, what happens when you give DLI? What happens after you apply DLI? And we saw that, first of all, there's a big expansion in the phenotypic diversity. And the responders, while they didn't quite have the response they wanted, dramatically expanded. They changed dramatically. They expanded much more than the responders expanded, even though after expansion, we still had higher diversity in the non-responders. So we see there's an expansion of phenotypes, and we wanted to ask, well, what are these populations that are expanding? Now, when we did it vanilla, we couldn't find any significant change. And that's because, again, we're dealing with these small, tiny clusters in these small, tiny samples, and actually quantifying the proportion of a cluster in single cell data is a very sensitive and noisy thing to do. And, you know, therefore, we decided to perform meta-clustering to sort of say, okay, but there might be some very similar clusters, and we can gain statistical power from merging them. And as you can see, you know, the bluer it is, the more similar it is. You can see a number of meta-clusters that are very similar, that have this very strong blue thing where, you know, these clusters are almost the same. So we merged them into meta-clusters, and indeed, once they were merged into meta-clusters, we could find that we could find significantly expanding clusters across patients that significantly expanded. And you know, these significantly expanded clusters actually expressed many genes of progenitor exhausted cells. And we found, you know, two such subsets of progenitor exhausted T cells that had both CD4 and CD8s in them. And again, some of these key markers, such as TCF7 and CXRCA5, were found previously to be important in response to therapy, at least TCF7 also in humans. So what we see is, you know, pre-DLI, we have, you know, an enrichment for terminally exhausted populations, but the populations that expand are actually not these, but these different set of clusters, these progenitor exhausted subsets. And, you know, these two distinct types, you know, it actually really matches mouse work that was done in mice where, you know, PD1 seems to exactly expand the PE clusters, really showing that DLI is, you know, can have a very similar immunotherapy mechanism where, you know, this reinvigoration happens through the expansion of these progenitor exhausted populations. You know, here we showed it in DLI in humans, and it was shown before really nicely in PD1 blockade in mouse. So, you know, we had this longitudinal data. We had more than 2 samples, and we wanted to see, you know, can we really dig into that? Can we use all these other samples? So, you know, our analysis to now was really focused on these 2 narrow slivers. And now we want to really capture the dynamics. We want to be able to ask questions of when and how and what are the dynamics and timing of these populations. So for this, we tried to build, you know, a model, a functional model of these populations, and really to compute a function that allows us to predict for any time point where one value predicts, you know, what is the structure, what is the timing of this population, really taking into account that because this is clinical data, we couldn't control for it like in mouse. We had, you know, very different time points, very different sample sizes, and a lot of challenges. So, you know, we built a Gaussian process, and a special Gaussian process that particularly took into account the fact that some of our samples had much fewer cells. We had a lot of dropouts, missing values. So we really had to take into account that not all samples have the same number of cells, not all samples are of the same quality, and we needed to sort of adjust our learning and, you know, trust more the high quality samples than the lower quality samples. And what we could get is we could get cluster dynamics, and we saw that the TE clusters really follow the dynamics of the tumor burden right here in the responders. You can see they going up and down. This gray line right here is the tumor burden. This blue line is the TE cluster, and you can see it really follows. And, you know, in the non-responders, it's doing whatever. The PE clusters, on the other hand, right after DLI therapy, both PE1 and PE2 began to expand, to dramatically expand right after therapy was given. And as these clusters expanded, the tumor was receding and tumor mass was getting smaller, whereas the progenitor exhausted in the non-responders weren't growing at all. They weren't expanding as the tumor continued to grow and grow and grow. So, you know, we see dynamics, we see these, you know, progenitor exhausted cells, we see all these differences between responders and non-responders and all these states and substates. So we said, okay, can we figure out some of the regulators underlying these things? Can we say, something about the regulators? And the question is, oops, I'm trying to look at some of the questions to see if there's some questions that really get people stuck and give the talk at once. And so, we also collected, the royal we being Pavan and Kathy's team, some, sorted ATAC-seq data, sorted for CD4, CD8, memory, and naive. So these weren't exactly sorted by our clusters, but they were sorted based on strong immunological cell types. And, we wanted to understand the transcription factors that might be underlying some of these differences, some of the epigenetic changes. And first, when we looked at the data, we got strong confirmation, and that there was very large-scale epigenetic remodeling between the responders and non-responders. The epigenetic landscape differed dramatically and differed in all the key, poster childs that you would expect, like TIM3 and PD1. And, these are just two examples, but they're two examples from a very largely remodeled landscape that's very different between, responders and non-responders, both before and after therapy. But we have many challenges here. For example, the bulk sorted populations don't exactly match our clusters. The binding factors don't exactly pop out of ATAC-seq data, even though there's some information about that. So here we needed to develop another model. There's a lot of models that sort of went into this work. And this is work by Cassandra and Elam again, and our sort of symphony multi-view mixture model. And the data that we had is our single cell observations, as well as bulk ATAC-seq data in replicates at least, which gave us a lot of statistical power here. And the idea is we wanted to sort of build a generative process that sort of explains regulation using this data. So we said, okay, what regulates is the open chromatin regions. So what we'd like is we'd like a different ATAC peaks for each population. And they have to combine to become the bulk ATAC peak that we see. But actually we have some information about the clusters and the cluster proportions from our single cell data, because if we cluster our single cell data, we know the clusters, we know their proportions, and we want these proportions to match. But we really connect the dot when we want to connect the dots through a regulatory model that says TFs should be regulating targets in particular ways, and that this gene regulation should explain the covariant patterns that we see in our data. So that the TF to target relationship should be informed but not constrained by the ATAC data that we see, and should explain the covariation data that we see. We're not limited to the things we see in the ATAC-seq, because there are many transcription factors, many key players where we don't have good binding motifs for. So if a good binding motif can explain a covariate relationship, we'd like to take it, but there are many factors where if we don't have a good motif and we have a TF to target relationship that we see in the covariate patterns or the data, we take that as well. And basically we try and combine everything to define a model that uses our observation to explain the data, and build a regulatory model that's supported both by a TF and a target having very strong covariate relationships, and preferably with accessibility tracks supporting a binding motif. So first I want to show you that this sort of works, so we can apply it to immune subsets that are known, sort of data that's more controlled. And here we sort of mixed a bunch of subsets that we knew. We mixed sorted populations of different immune clusters. So we knew what went in, so we had a ground truth, and we could see how well did Symphony do the deconvolution. And we saw that Symphony's deconvolution was very accurate relative to other deconvolution algorithms at the time. Again, a ground truth for TF to target relationships is very hard to find, but we used some data with perturbations to see that we got pretty accurate readings of TFs to targets, especially since this is a noisy and rife with false positive and negative ground truth to begin with. So with a little bit of confidence that what we got is at least in some of our test cases working quite well. And again, my favorite test case of them all, the algorithm is very robust. We applied it to all the data that we'd collected together with Kathy and Pavan. And we saw that indeed we could get a map of what are the key regulators that are driving each of these clusters. And while we tried to infer these regulators across multiple samples and across multiple clusters, what we could see is that there were a set of regulators that repeatedly were selected across the different TE clusters. We saw a structure of sort of master regulators that could explain many of the differences that we saw in the TE clusters. And we saw a set of regulators for PE1, PE2, again, shared across the clusters that participated in each group, as well as a bunch of shared. And what are these master regulators? They are regulators that could explain the differences that we see among the differentially expressed genes that sort of define each and every cluster. And among this, and this is sort of unbiased de novo analysis that didn't take into account any prior knowledge, we see TOX coming up in the terminally exhausted. We see TCSF coming up in the progenitor exhausted. And, we even see some factors that were found by Merrimet, GATA3, and MTF2 as part of the terminally exhausted. And one of the things that we found is, again, we found two different PE clusters that are driven by two different regulators. Zooming in, we could actually, zoom into each, to see what are the regulatory networks that we see for each of these clusters. You could see the correlation and the covariate structures underlying each of these edges. And, we see that many of the master regulators, such as TOX, are the master regulators that sort of control PD1, PRF1, TCF7, controls all sorts of key markers, including in a feedback loop itself. And we really see a nice network architecture, and we're still digging into these things. The final thing that sort of we want to look at is, are these progenitor subsets that are expanding, that are really growing, that are really leading to this response, are they introduced by the actual DLI? We are giving donor lymphocytes in. So first of all, we look at the TEPE relations and see that they're clonally related, that the progenitor exhausted and the terminal exhaustants often share the same clonotype. So really it is the progenitor exhausted that are sometimes, leading later on to terminally exhausted cells. They have a shared lineage. But what was particularly exciting is that a lot of the clonal expansion, these blue lines, these were actually TCR clonotypes that existed in the patient prior to therapy. So they were there, and something in the DLI product was reinvigorating. Most of the, there's some things that, was shared with the DLI product, post, 4 weeks later, and we don't know if it was there in small abundance or we didn't pick it up or if they're new. But what we found is that there was almost no clonotypes that were expanded that came from the DLI product themselves. It was either sort of new clonotypes that we identified, and the biggest expanders were there identified pre-DLI, and sometimes we could even see them, years later. And we had similar dynamics for an additional patient. So really, the model that we get here is that after DLI, we have these progenitor exhausted cells that reinvigorate and expand to create the response. Whereas the non-responders have, distinct states of dysfunction. There's no one-stop solution that will give a solution to all patients. Each patient has its own dysfunctionality. And again, I think, I've said these conclusions a few times, so we can skip them. I had another story, but of course that was a little bit, ambitious of me. So I'm just gonna skip the story because it's been a long talk already. To take home points that, tumors and their immune systems dynamically remodel. We need methods to get it. Computational choices really matter, interpret with care, and we're still missing a lot of methods. And again, acknowledgements. This has been a wonderful and fun collaboration with Kathy and Pavan. I've talked a lot of work with together with Sasha Rudensky, where we did our initial forays into tumor immune microenvironments, and some algorithmic work that was done with many members of my lab. I also mentioned some work with Scott Lowe, and thank you very much for your attention.
Okay, thanks, Dana, for the great presentation, and we have a number of questions. Some of them relate more to biology, others more to computation. So we try to answer a few of them at least. Sachet is asking how can you explain, like, for looking for common factors across patients and cells help you identify transcription drivers of responders versus non-responders?
I didn't quite understand the question. So what?
Could you explain how looking for common factors across patients and cells help you identify transcription drivers of responders versus non-responders?
So those were two different computations. So the computations were basically, you take the data. So one of the reasons that some of the early steps in data processing and normalization and batch correction matter is that, once you've applied these to the data, then all your downstream analysis follows from that. So I tried to sort of really put emphasis on some of those early steps because, once you've sort of settled on your count matrix and normalize it, then you apply a lot of different methods to that. After that, almost every method that I described here was a separate analysis. The volume analysis was applied separately. The common factor analysis was applied separately. And it found a set of common factors. I looked at what those common factors, well, I didn't do anything. Elham and Pavin did everything. Elham and Pavin looked at what these common factors might be. And, the symphony model is a completely different model. So the symphony model went back to the raw counts, and the symphony model integrated in the ATAC-seq data and didn't use the common factor analysis at all in its analysis. So those were it's just sort of a toolbox of different approaches to try and elucidate understanding from your data. And each one of these tools are separate and separately applied.
Okay, a related question to the last part of the talk. Was there reversal of exhaustion in the PE clusters 1 and 2 when they were expanding and the tumor burden was decreasing in a patient?
So again, I think that these PE clusters themselves were expanding and not necessarily reversing, but that sort of they fed maybe other factors that didn't. These are sort of progenitors that create new clusters. So those clusters themselves were fairly similar, and I'd have to look if I didn't look to see if those were reversed or not. But what I would assume is that, and again, this is assumption, and I didn't quite look at that at the data, is that they are the progenitors that sort of generate, I guess, T cells that aren't exhausted if we see this reversal. But I have to check that. Again, the fact that they are so similar, it sort of depends on what, I didn't check that particular thing.
Okay, then some more computational questions. How do you compute covariance? Is this just correlation between gene expression? I think that could be a
no. So covariance is not correlation, because correlation really looks at each pair of gene expression. Covariance is really it's the second moment of the data. So what we really have is a high dimensional Gaussian. And we take the sort of full dimensionality of the data into account. That's one of the reasons why covariance is so expensive, because you are treating this as a multivariate, multidimensional Gaussian, and you're computing the second moment of that in very high dimension, creating a pretty heavy computation. But there's statistical, covariance is I didn't invent that. It's the covariance of a multivariate Gaussian is something that any statistical toolbox will give you an option to compute through heavy matrix computation. It will just be heavy. One of the reasons covariance isn't used as much as it could and should be, and even ourselves that we see its power don't use it that frequently, is it is computationally heavy, and I'm going to put 3 exclamation points after that one about the heaviness of its computation.
So another question is actually 2 questions related to BiSKIT. are you making this available on the cloud for general use? And if not, what are the suggested alternatives?
Yeah, so again, I sort of mentioned that as I was reading the questions while actually giving the talk. That's when you saw me seem semi-distracted. So we didn't make it available on the cloud, but we do have it. You know, there's links to it, I believe, from the Aziziel cell paper. I believe it's on my GitHub. Now, it is a complicated algorithm to run. It takes a little bit of tuning of the parameters. It takes a little bit of, it's not a plug and play Seurat-like algorithm, and requires a little bit of expertise to run, but it is available. It's just hard to make an algorithm like that incredibly friendly. Again, Biscuit has sort of two features. One of them is really takes into account the fact that you want to have different normalization for the different cell types, and I think that's particularly important in TILs. And John Marioni's SCRAN, as I've mentioned earlier, does that as well, and we've seen, you know, very good results with the much less complex and heavy SCRAN with complex tissues that have very many different cell types. Mm-hmm. And Biscuit itself is, it really is, you know, we've had very good results with it. And sometimes when we think we really need it, nothing replaces it. But it's heavy and requires some understanding when you're trying to use it. But it is available, just not easy to use.
Okay, thank you very much. So unfortunately, we don't have time to answer more questions. We will have Dana have access to these, and then she can get back to others. So thank you again for the very nice talk, and thank you for the audience for listening. And again, a reminder that the slides will be posted on the website later today, so you can look through them in case you could not catch everything. Thank you.